So I'm presenting ESR number 4, Nikolaos Katsinas. He comes from the University of Valladolid here in Spain, and his supervisors are Dr. Soraya Rodriguez Rojo and myself. He is from Greece. He has a bachelor and a master degree in pharmacy and master of science degree in natural product chemistry. And the title of his presentation is Valorization of the olive byproducts phenolic compounds and their evaluation as therapeutic agents for ocular surface inflammatory diseases. So Nikos, the floor is yours.
Thank you, Amalia, for presenting me. Good morning to all of you. Dry eye disease is a multifactorial disease in which inflammation and oxidative stress in the lacrimal functional unit create an abnormal tear film composition and thus an unstable precorneal film layer. Nowadays, the most important and the most effective therapies used are anti-inflammatory compounds that target the vicious circle of this disease, of the inflammation of this disease. So we mostly use topical steroids. They are widely used. However, they cannot be used long-term because they create several complications such as infections, cataract, or glaucoma. Cyclosporine in 2003 was the first molecule that was approved as a dry eye disease therapy. And however, it needs several months to achieve a therapeutic effect. And also, it has a very low solubility in water, and that's why emulsions were generated in order to increase its solubility in water. However, the oils and superfactants present in these emulsions are really irritating for the eye, creating even blurry vision in some cases. The latest molecule approved for dry eye disease was lefitegrast, also acting by decreasing the migration of lymphocytes and the cytokines secretion. However, also it has some side effects such as decrease of visual acuity or dysgeusia, and also it's only approved in 6 countries, still not by the EMA. It's not approved by the EMA, so we can clearly see that there is scarcity of effective therapy for dry eye disease. So in the search of new therapies, the phenolic compounds were proposed as a new therapeutic alternative due to their versatile biological activities. Actually, and among them, the olivephenolic compounds have been widely reported because of also their widely known biological activities. We can see the importance of the phenolic compound back in the history. Even in the ancient time, in ancient Greece, the olive tree was used in the everyday life of the ancient Greeks. And nowadays we know that the importance, even today we all know the importance of the olive oil in the healthy Mediterranean diet. And now we know that the olive oil, it's based, the importance of the olive oil in our everyday life is based on the olivephenolic compounds who are present in it, and they have numerous biological activities, and among them antioxidant and anti-inflammatory. The most reported compounds, olivephenolic compounds, are oleuropein and hydroxytyrosol. Currently, through the industrial olive oil production, the mostly used nowadays, 4 principal byproducts are produced. The pits, the twigs and leaves, the wastewaters, and the olive pomace. Actually, the byproducts produced are produced as 4 times more than the olive oil itself produced. And among them, the olive pomace is the principal byproduct. It is produced actually in huge quantities, between 7 and 30 million cubic meters only in the Mediterranean area. And it's actually the semi-solid byproduct. It's a mixture of vegetation and processed waters, smashed pits and pulp. And also it's important to highlight that 80% of the olive fruit ends to be olive pomace. Only 20% becomes an olive oil. And similarly, 80% of the olive fruit phenolic compounds end up to the olive pomace. Only 20% go to the olive oil. So it's important to see that these compounds give to this byproduct high organic load and phenolic content, which increase the biological and chemical demand for oxygen, and also give to this byproduct phytotoxic and antimicrobial properties. Therefore, a very important environmental concern is generated, and several valorization strategies are being performed. So our objective was to optimize the phenolic recovery from this byproduct, from the olive pomace, then use crude phenolic olive pomace extract, as well as the pure compounds hydroxytyrosol and olivropane in vitro on human ocular surface cells to test their in vitro antioxidant, anti-inflammatory, and immunosuppressive activity, then select according to the best in vitro performance, the best agents to test their in vivo anti-inflammatory effect in a mouse dry eye model, and finally evaluate both compounds and the olive pomace extracts as future ophthalmic products. So first, regarding the optimization of phenolic extraction, we tested the best storage condition of the material regarding the stability of the compounds present in it. Then also we tested different defatting pretreatments in order to reduce the residual olive oil from the byproduct. The residual olive oil, it's calculated around 2 to 3%. This was done actually to reduce the irritation for the eye, as we previously mentioned, but also by the defatting pretreatment, we increase the subsequent phenolic recovery. And finally, of course, to do the phenolic extraction, to select the best method and optimize the method through statistical model, finally, to select the most promising extracts. So among the storage conditions, the lyophilization was found to be the best regarding the stability of the compounds in it. And so using conventional extraction methods, we generated a conventional extract, hereby conventional, and it was used as reference for our subsequent in vitro studies. Then, between the defatting pretreatment methods, a conventional one using organic solvent and the supercritical carbon dioxide were tested. The conventional is the principally used nowadays in the industry, and so between these two, the supercritical carbon dioxide was selected because the yield of the olive oil was similar in both cases, and also toxic organic solvents are avoided with this method, so it's an eco-friendly method. And finally, the pressurized liquid extraction was selected for the phenolic extraction, and a design of experiment was applied to this method, which is actually a statistical model that with this model, we can optimize the extraction parameter, the experimental, let's say, parameter in general, through a small number of experiments. The model used was the central composite design, which was based on the bibliography because of its better predictability. And so a sequential extraction method was established, in which actually that means that in different extraction conditions, we were able to optimize the phenolic extraction differently for each compound. So we selected 3 different extraction conditions, giving birth to 3 different extracts. The optimized extract 1 that had the optimal chemical antioxidant activity, the optimized extract 2 with the highest concentration for olivropane, and the 3 with the highest concentration in hydroxytyrosol. This work was published in ACS Sustainable Chemistry and Engineering in April of this year. So then moving up to the in vitro studies, first we established the maximum allowable concentrations of all treatments of these 4 extracts, as well as the pure hydroxytyrosol and olivropane on human corneal and conjunctival epithelial cells. And then we did firstly a screening of the 4 extracts, the 3 optimized and the conventional one, on corneal cells under the same concentrations. Then we selected 2 of them, and together with the pure compounds, we moved to dose-dependent studies on human corneal and conjunctival epithelial cells. We also tested low-dose mixtures of the pure compounds to test the synergistic effect on both cell lines. And finally, we tested also the inhibition of proliferation of human CD4 cells by the selected treatments. So regarding the in vitro studies on corneal cells, for the antioxidant activity, actually here it should be here, the circle, as far as I see now. The optimized 2 and 3 were the 2 extracts that had the highest antioxidant activity. The 2 is the optimal for olivropane and the 3 for hydroxytyrosol. Whereas regarding anti-inflammatory, the optimized 3 and the conventional had the optimal anti-inflammatory activity. So the conventional and the optimized 3 were selected to move forward to the dose-dependent studies. So in corneal cells, we can see that all treatments, pure compounds and extract, had a really strong antioxidant effect, a synergistic effect also for the low-dose mixture. And for the anti-inflammatory, although olivropane did not have any anti-inflammatory effect, both extracts and the pure hydroxytyrosol did have anti-inflammatory effect from low concentrations. Also, we detected a synergistic effect for the IP-10. It's important also to highlight that the optimized extract in general was active at 2 to 40 times lower concentration compared to the conventional one. Similarly to the conjunctival cells, all treatments had a very good antioxidant activity, but here we had activity only for the IP-10 molecule, but it's important to see that here olivropane also had an anti-inflammatory effect. And also here, similarly, the optimized extract was active at 10 to 40 times lower concentrations. This work was published at the Journal Antioxidants in July of this year, and a patent was submitted at the Spanish Patent Office. Closing the in vitro studies, the inhibition of proliferation of human CD40 cells isolated from peripheral blood was tested for the selected treatments now. We selected the hydroxytyrosol and also the optimized extract among the 4 treatments, the 5 actually treatments. And among the concentrations tested, the highest concentration for hydroxytyrosol and 2 out of 3 for the extract were found to have an immunosuppressive effect. Moving forward, for these 2 selected treatments, we moved to in vivo studies in a mouse dry eye model of desiccating stress and scopolamine administration. Actually, we did 2 experimental sets of 14 days each one, compared to the vehicle-treated group, these 2 treatments, which was borate buffer. And we measured tear production, corneal fluorescence staining score to measure the corneal damage, lymphocyte levels in bicervical lymph nodes, and cytokine gene expression in the cornea, conjunctiva, and lacrimal glands. Regarding tear production, although it is important to see that the tear production was reduced significantly after 11 days of desiccating stress and scopolamine administration. However, it's important to see that for optimized 3, for the extract, the decrease after 11 days was not considered statistically significant. However, it's also true that none of the treatments improved the tear production after 11 days of desiccating stress. For the corneal fluorescence staining, both treatments, however, were really effective in reducing the fluorescence staining score after 11 days of desiccating stress. And regarding lymphocytes levels in cervical lymph nodes, hydroxytyrosol decreased cell count of CD3 cells, whereas the extract decreased CD8 alpha cell count. Regarding the IP-10 gene expression in the tissues of the lacrimal functional unit, IP-10 gene expression was decreased by the extract in the conjunctiva and the lacrimal glands, and by hydroxytyrosol in the lacrimal glands. For the TNF-alpha, the gene expression of TNF-alpha was reduced in the cornea by the optimized extract. This work now is under preparation, more correctly is prepared, but it will be submitted shortly. And finally, after all these in vivo and in vitro studies, we moved to the evaluation of future ophthalmic compounds of the 2 selected extracts and the 2 compounds, the pure compounds. Firstly, we had to evaluate the genotoxic effect on human corneal conjunctival cells of the treatments. Also, we wanted to evaluate the long-term stability of the aqueous solution of the treatments, and move forward to putatively identify possible degradation byproducts of the compound and compare the degradation profile of the 2 compounds in pure or extracted solution. Finally, we wanted to establish a baseline for the analysis protocol of these products in the future, because it's important to see that there is no monograph for these, neither for the compounds nor for the extracts in the pharmacopoeias. So it would be a very important baseline for the future evaluation, industrial evaluation of these treatments. So regarding the in vitro genotoxicity, no genotoxic no genotoxic effect was detected for none of the treatments, because none of the treatments had any statistically significant difference between any treatment with the control group. Whereas now regarding the long-term stability studies, the first month, we can see clearly that we have, for both compounds, there was a different stability in pure or extract solution. We can see, for example, that hydroxytyrosol increases in the extract while decreases in the extract solution. So for this reason, we collaborated with EuDES to putatively identify and see the degradation profile of these products. And also, we confirmed that olivropane is transformed into hydroxytyrosol in the extract, but not in the pure solution. And yes, we identified the byproducts of olivropane. However, we were not able to identify the byproducts of hydroxytyrosol. The results after the first month, up to 6 months, actually confirmed the result of the first month. This work was also published in the journal Molecules in October of this year. So finally, we can say that storage and pretreatment conditions were established for the raw material to enhance the stability of the compounds. Then we also established a sustainable, selective, scalable, and scalable sequential extraction process able to increase and optimize the richness of the extract obtained in the compounds of interest, and actually increase it by 2 to 6 times depending on the compound, always compared to the conventional methods. All treatments were able to palliate the response to oxidative stress of the human ocular surface epithelium. Also, for both extracts and hydroxytyrosol, they were both able to, all the three were able to palliate the innate inflammatory response of the human ocular surface epithelium, whereas hydroxytyrosol and the optimized extract were able to also be used as therapy for immune-based inflammatory pathologies and also for the treatment of dry eye disease. All treatments can be safely used in the ocular surface, and also a necessary evaluation baseline was established for the future approval of these treatments as ophthalmic products. So, always is what's next. First of all, we can say that the one important step would be the scale-up studies of the extraction process in order to verify that there is a scalability in our extraction process established, and see if the optimization and the obtention is similar, is maintained at a large scale. Of course, as I previously mentioned, the byproducts of hydroxytyrosol were not identified with the method used, so we need to test different methods to be able to identify the byproducts of hydroxytyrosol. Also, although we established the evaluation of degradation byproducts, however, none of the treatments, the aqueous solution, fulfilled the requirements of the guidelines of the 95% concentration, so therefore it's normal because it's not a final formulated product. So of course we need to follow up with future formulation studies to increase the stability. Apart from genotoxicity assay, we need to do also irritation tests for the stability, for the approval of the products. During my secondment in EuDES, we started also a study of purification that moved forward and had a nice results to increase the extract richness in the compounds of interest. However, it was interrupted due to COVID outbreak, so one of the things that also need to be completed is this project. And of course, the objective of all discoveries always is to bring from the bench to the bedside discoveries. This is the objective of EuDES. So the big objective would be, the biggest future study of this would be to test clinically these treatments. I want to thank a lot of people, a lot of groups, the IOBA group, the High Pressure Processes group, EuDES, University of Cologne. From names, I'm going to say only Soraya and Amalia, and a lot of people, and of course, all of you who were here and listened to me.
Thank you, Nikos. No time for questions.
Hi.
Hi.
Can you hear me? Is this thing on? Yes. Okay, good. Really good talk, Nikos. I noticed you used some very specific words, which I think are interesting. You mentioned that these things needed to be monographed. And at least in the United States, what that means to people is that if it's monographed, they can use it as an excipient in a vehicle without any type of regulatory interference. But at the same time, you're showing a lot of data that indicates that these things are anti-inflammatory, et cetera. The FDA would look at that, look at those as actives, right? I mean, and those things would have to go through complete drug evaluations and clinical trials. So what's the goal here? I mean, the work that you and Amalia and Soraya have done is amazing, but what's the goal? To make a formulation vehicle or to actually make a drug?
But to make a drug, we need a formulation, no? To increase the stability.
Right, but right.
I'm not sure if I understood well.
No,
the
you need a
question.
formulation for sure.
Yeah,
But what you put in that formulation is critical.
of course.
So are these allophenolic compounds considered drugs? Or are they just a
Yes.
You use the word palliative a lot, which is a great word to use if you just want to use something as like an artificial tear. Palliates the ocular surface.
No, actually we can say that we're going to be used as a drug, but this
Okay.
means a lot of regulatory
Right, you're going to have to run
procedures.
trials with those.
Yes.
Yeah, that's what I was getting at.
Yes. First of all, the first step would be to identify for hydroxytyrosol the byproduct. So we need a lot of studies in this analytically in this field to be able to see all the byproducts. To actually also, since it's a plant material, it needs to have some for microbial properties, etc. So it needs to be very specific, everything. So it needs a lot of studies in this field.
Congratulations for your talk. Very, very nice talk.
Thank you.
What is the question raised by Mike is critical, because effectively there are two strategies. One simple, it's a medical device with no major pharmacodynamics or pharmacological issues, and the other one is it's truly a medication. In that case, of course, it takes a lot of time. But regarding antioxidative drugs, we are at the frontier. And I know that in Europe, in EMA, or at least France, accepted antioxidative agents and still keeping them in the category of medical devices. So maybe it's a way to go fast to registration. And but this problem is, in turn, if it makes facilitate the approval, at the end it's very difficult to communicate on pharmacodynamic properties. And it's the case for hyaluronic acid, for example. Hyaluronic acid is approved and well developed in Europe, but it's considered as a medical device. So there is no major problem. But the companies are extremely scared about the fact that if somebody communicates on anti-inflammatory properties or wound healing properties or other properties of hyaluronic acid, they will be told, oh, you are telling us that your medical device is not neutral, but it is pharmacologically active. So go get out of the medical device category and come back in 10 years when you have enough data to prove that you have a true medication. So of course it's a
big topic.
It's difficult.
Yes, the regulations.
And just not to be too long, another question, but maybe at that step it's too early to have the answer. Sometimes lipidic compound or other antioxidative agents may be not extremely well tolerated. This may be an issue, especially because those patients who will be the good patient for this category of compound have extremely sensitive eyes. So sometimes lipids are not extremely well tolerated. We know that with cyclosporine, for example, but with other lipidic compounds. This is not to prevent you to continue.
Compound,
Sorry,
the lipidic compound.
but it's not exactly.
But exactly what we wanted to do is this, to reduce, that's why we did the defatting pretreatment, reduce and remove any lipidic compounds present.
Good.
We worked with phenolic compounds that are medium polarity compounds, So it's a different category. We didn't want to have lipids because of this. From the beginning, we had this target.
Thank you.
Yes, congratulations. Very nice talk.
Thank you.
Maybe a comment and a question. A comment is I know that for medical devices, the regulations are also getting more strict each year. So the difference between a drug and a medical device is becoming less and less. So I don't know if it will be important for you, but it will be challenging in both cases, I think. So that's a small comment that I wanted to make. And for your extract, it's a question. So how will you ensure that each batch that you produce is in fact of the same quality? I think it's a... what are the constituents? Which will you follow?
Huge question. Huge question, huge future study also for us, because we need to, first of all, end up with among the different cultivars, because we used our Bekina variety and there are several. We need also to test different varieties. Then we need to test the period that we're gonna of the harvest. So that's a different study also as well. So finally, I think that all medicines that include extracts go with a concentration of the active ingredient in it. So it needs to have a specific concentration, the extract, like among this range.
Yeah, and which one will you choose? Because it has many active ingredients.
I think hydroxyrosol, because actually the extract that we used, that had the best activity and it had a really good in vivo performance, was the optimized for hydroxyrosol. So they went together, hydroxyrosol as pure compound and the extract with the highest, and enriched in hydroxyrosol.
And a small question is, what did you use as a readout for oxidative stress?
Intracellular ROS.
And how do you measure? How did you measure?
With the H2DCFDA dye assay, through the UV, through after 15 seconds exposure to UVB rays.
Okay.
Okay. We have a question online from Philip. And I'm reading
Hi,
it.
Philip.
It says, great work, Nikos. Have you thought about forming a startup collaboration with EuDES?
A startup collaboration? No. No, I haven't received any proposal. Philip? Can you hear me? No, so far nothing.
No.
We still have some months left to talk about this.
Okay, I don't know if there is any other question in the room.
I think we have one more here, Amalia.
Okay.
Just a short comment. Do you expect to have a metabolization of your compound according to you know all the enzymes in the tears in cells? Do you expect to have metabolization of this compound, and if the metabolite could be toxic or no? Just a naive question.
Yes, I think that since the blink is really fast and the contact with the eye during blinking is really, so the contact is really short, I think there is not enough time of contact and with this base, we didn't think this part of studies of seeing the metabolic byproducts, if they are toxic or not, but it would be interesting to see it.
It's perhaps indirect question. Can we check the reliable enzyme to cut this compound?
We could check it, yes, but we had not thought about it, I'm honest.
Okay, thank you.
But yes, if we check it something like this, we have to do the reaction with this with the enzymes of the ocular surface to see which byproducts are produced and then see if they are toxic or not. Yes, it would be really interesting as well. I think not.
Oh, okay. So thank you, Nikos.